Biology. Today we'll deal with the topic of karyotyping. We have already dealt with the types of banding pattern, and based on banding pattern, this technique of karyotyping has been developed. So what is karyotyping? Karyotyping is a laboratory technique which is used to visualize and identify the number, size, shapes and banding pattern of the chromosomes. So for studying the patterns of chromosomes, the size and the number karyotype is being used. And the image which we get after studying or after observing the banding pattern, that image is known as karyotype. Karyo means nucleus and karyotyping is basically the classification of nuclear chromosomes. We know that the chromosomes can be stained. I have already talked about 6 types of banding patterns which we can observe in chromosome, and all those can be classified. When we match the chromosomes of two persons, the number and the size of the chromosomes will be similar, banding pattern will be similar, but it is not exactly 100% matching. So what is the purpose of doing karyotyping? We can detect the genetic disorders. So genetic disorders can be detected. The sex can be determined whether it will be a male or will be a female, because when you study the karyotype, you will find that the sex chromosomes are all both are XX or XY. It will easily be observed, so you can determine the sex of the child. Cancer diagnosis is another event where if there is some translocation of portion of gene, or there is some abnormalities, there is deletion of portion, then that chromosome will not match with the standard. It means that there is some changes in the structure of the chromosome which are at 1% chance to cause cancer. Prenatal screening: the fetal chromosomes can be screened from the amniotic fluid for checking the genetic disorders. Species comparisons: one species, when we want to create an evolutionary relationship, we can go for species comparison. Fertility issues: we can detect the chromosomes which cause the infertility and miscarriages. We can correct it with the help of gene therapy. So this is the main focus why karyotyping has been studied, or this technique has been developed, because it has so many numbers of positive results which you can obtain. Now, what is the procedure? First step is sample collection. In this, the sample can be blood, amniotic fluid, bone marrow, or a tissue. This anything can be preferred. White blood cells, lymphocytes are also preferred. So, it is the first step is sample collection. Then the second step is to culture the cell. So being an animal cells, we have to go with animal cell culturing mediums, the nutrients and the mitogens, as well as we should provide it with a good temperatures and incubation conditions, so that the cells can be cultured easily. Then the next step is we have to arrest all the cells in metaphase, because in the banding pattern also I have explained that the banding patterns are studied in metaphase state only. So all the cells has to be arrested to a metaphase, and this can be done by adding colchicine. So when we are doing the cell culture, after few generations or few hours, we will add colchicine to the medium so that the chromosomes will get arrested. After getting all the cells in metaphase, then we have to treat it with a hypotonic treatment. Hypotonic treatment are basically we use KCl. Now this will cause swelling and the chromosomes will spread. Remember, we are talking about the cells. The cells are in metaphasic state. Inside the cell membrane. Now we are dipping the cells in KCl so that the cell will swell. As the cell will swell, then it will get much more area for the chromosomes to be spreaded. And when it is spreaded, then it is easy to visualize. So the step is to easily visualize the chromosomes. Now we move to fixation. Now, when the cells have been swelled and the chromosomes are in spreaded condition, we have to fix the cell in that position only. And when we want to fix it, we will add methanol to acetic acid. The ratio is 3:1. And when we add it in this ratio, then the position of the chromosomes will be fixed. Now we'll drop a cell suspension on a glass slide and then it will stain with gypsum. Our other banding patterns, we have dealt with 6 banding patterns. If you have not seen the videos, you can go through the content and visualize the banding patterns of chromosomes. So any of the banding pattern, whichever you are required for. If you want to focus the centromeres, you can go for C banding. If you normally want, you can go for G banding. After this staining, we can observe this in microscope and then we can take a photograph. After taking a photograph, all the chromosomes has to be arranged in pairs and then we will get that karyotype. Now, what features are analyzed in a karyotype? After getting that analyzed report, what we will understand? You can get the total number of chromosomes. You can see the sex chromosomes, whatever it is present. You can study the chromosome morphology. You can see the position of centromere, the length of the centromere, the length of the chromosomes. Either both the P and Q arms are equal, or there is a chromosome position is at center, or there it is near to the end. Whatever it is, we have to, you can study. You can study the bending patterns, and the most important thing is, with while we study all these, we can get if there is some abnormalities like deletion, duplication, inversion, translocation of ring of chromosome. If all these have happened in the chromosome, then the banding pattern, when you visualize the chromosome, you will find that the length of the chromosome is short, or it is long, or there is a abnormal changes in the banding patterns. All this will specify that yes, there is some abnormality in the structure of DNA. Next is types of chromosomal abnormalities. There are numerical changes. The number of chromosomes can be changed. If there is addition of one extra chromosome, then it is trisomy. If there is missing of one chromosome, then it is monosomy. If there is whole set of extra chromosome, then it is triploidy, because diploid is natural. If the extra set is again present, one more set is present, then it is triploid. If two sets are present, then it is tetraploid. So this is the change in the number of chromosomal abnormalities. There can be changes in the structure of chromosomal abnormalities. There can be deletion of portions of the chromosomes. There can be duplication of the chromosome. There can be inversions, translocation. Inversion is orientation of the DNA in reverse direction. If it is 1, 2, 3, then it will be 3, 2, 1. Translocation is part of chromosome separating from one chromosome and moving to another chromosome. Ring structure is when the ends of the chromosome get fused. Normally, when we talk about a chromosome structure, it has telomeres at the end, and telomeres have the tendency not to attach with other chromosomes. But if that property is missing, then the chromosomes can form a ring structure. Now, when we talk about human karyotype, we will find that total chromosome is 46, 23 pairs, 22 are autosomes, and one pair is sex chromosome. Either it will be XX or XY. The normal female has 46 with XX, and normal male will have 46 with X and Y. Now, how to learn that? The steps you can learn by remembering the sentence, cool human fight, stained monster. Cool is for, from cool you can get C, that is culturing in hypotonic solution. Then we have to get it fixed, then we have to stain, and again we have to microscope. So first one is isolation of the cell, then we have cell culturing. So C stands for culturing. After culturing we have to treat it with hypotonic solution so that the cell will spread, swell, and the chromosome will spread. We can use KCl, so it is hypotonic treatment. Then we have to fix it. Now for fixing it, we will use some of the stains what is required. We will use methanol with acetic acid, so it will get fixed at that metaphase only. And then we have to go for staining, and we can go for Giemsa stainings or C staining, R staining as what required. Then we can observe it under microscope. Now, when we talk about the applications, it is medical genetics. It has a huge applications in obstetrics. When we talk about prenatal diagnosis in late pregnancies, where there are chances of chromosomal abnormalities in the fetus or in the newborn, then before the birth, at the age of 6 months to 8 months, there is diagnosis of the child. Then oncology, we can detect the tumor specific markers. In forensic science, we can use it for determination of sex chromosomes and the sex of the fetus as well as the human beings. In cases of some damages, some accidents, all that, we can use this. Animal breeding, the studies of the genetic traits is widely appreciated by the help of karyotyping. So these are the applications. Now, what is the limitation? You cannot detect a single gene mutation because we are talking about the whole chromosome. If there is a slight 1 micrometer change in the chromosomes, that could not be detected, and it requires a dividing cells. You cannot work on a dead cells, and some structural defects can also be undetected because uh, if there is translocation, that much portion must have a, the region which has been translocated must have a banding regions, and it is a time consuming experiment. You have to repeat it within duplicates or triplicates so that there is no false result in the karyotype. Now, when we talk about karyotyping and banding patterns, if you compare it, then the karyotyping is chromosome count and structures, whereas banding pattern is the detail of one's chromosome. We can use microscope and image processing. In banding pattern, we use specific strains. Karyotyping gives large abnormalities and number changes, whereas banding patterns are more specific. They give specificity of gene mapping as well as fine structures demonstration. So, for getting a karyotype, you have to follow the bandings, and after getting the banding patterns, only you can determine a karyotype. The process is known as karyotyping, and the result or the photographs what we is called is karyotype. So finally, we can conclude that karyotyping is a foundational techniques in genetics and clinical diagnosis. You have understood well that it is an important part in the medicinal science as well as in evolution also. If we have identified anything which is new to us, so we can go for evolutionary diagnosis also. Hope you will this will find you well. You all are being with us, so try to share this with your friends who requires this topics, with your classmates, so that you all can understand it. If you have any queries, you can give it in the comment section. If you need any specific topic to be explained, you can also give that also in the comment section. Thank you and happy studying.